Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Okay. It's my pleasure to introduce Yosef Barash. He's visiting us from Hebrew University, where he's about to receive his PhD. He started with a bachelor's degree in physics and computer science and art, and then went on to get his PhD at the Hebrew University near Friedman Street. Yosef?
Thank you very much. Okay. So what I'm going to talk about today is a flexible probabilistic framework to infer regulatory mechanisms. I'll start with a short bio-background. The central dogma as it comes to biology is that we have the DNA, the coding sequence, and then the coding DNA is transcribed into messenger RNA. This is done by the polymerase complex you see on top here. And then the mRNA is translated into basically into proteins which do whatever the cell needs them to do at the specific conditions that it is at. So that's the way that the cell can move from one condition to another and then control the different conditions that it is given. Now, one of the important things that we have is about these processes is we have measurements done at this exact stage that you see here, the mRNA. This is what we call gene expression measurements. Usually it would look like this. What you see here is that the columns are the experiments, and in each experiment you get to measure the mRNA levels of thousands of genes simultaneously. These are noisy measurements. Red here, the color scheme is that red represent overexpression and green represent underexpression. This is like basic gene expression slide that you usually see. It's important. And of course, to understand how the gene expression is regulated is a central biological research question. Because one of the major ways that the cell controls its behavior is by controlling which gene to produce at which stage, by controlling the production of the mRNA here. Okay, so just let's zoom in on this process over here. The way that the classical model of how the cell usually does this control at the transcription level is that you have what you have here is the gene coding area, which you see here as pink, and you have a non-coding area, which is called the promoter area. And you have different molecules called transcription factors or regulatory elements, which bind directly or via a mediator to small subsequences along these promoters. And by doing so, they affect the transcription of the nearby gene. They can initiate the process, they can block it, enhance, repress it, and so on and so on. And different combinations of different transcription factors are used for different cellular conditions to affect a subset of genes which the cell would need to be upregulated or so on. Like, for example, if you have stress conditions and you have high salt and the cell wall is damaged, you would need certain proteins to be produced very quickly, and you would use a certain combination of transcription factors to produce them, to enhance their production. Okay. Now, another important measurement that we sometimes have, if we're lucky, is called location data, which is like trying to measure the amount of binding between these proteins, these transcription factors, to the actual promoters. So what we have is transcription factors are the columns, the genes, again, are the rows. So these binding events would be these specific colors here that you see, these specific rubrics in the matrix. And the brightness conveys the amount that we are sure, or we believe that there is actual binding over there. Okay. So what we're actually interested in doing is we have like the general scheme is like we have this is the yeast cell. We observe different phenomena of it. We want to explain what is going on. So we take different measurements. That's the sequence of the yeast, which we already know. Yeast was already sequenced. We have the gene expression. We have the location. And we're trying to come up from them with some model of the processes that are going on in the living cell. And what we're interested in is regulation mechanisms that I've just described. So basically what we have is like the input would be the promoter regions, the sequences, and the gene expression data. And what we're trying to come up with is something like this. Now you see that the gene expression arrays are reordered. And we have here like a small subgroup of the genes, which are the rows, which are all overexpressed, this is red, at certain experiments, which happens to be all related to the G1 cell cycle phase in which they were done. And you can also see that all of them are, they have a specific motif in the promoter regions of transcription factor 1 for that example. So transcription factor 1 can be an explanation for the co-regulation, the upregulation at G1 cell cycle phase. But you also see another subset of them, which are all downregulated at G1, G2 cell cycle phase, and they all share another motif, which is the transcription factor number 2. So a combination of transcription factor 1 and 2 can explain the downregulation at cell cycle phase G2. So if to generalize what we have just described, we're trying to come up with these combinations of which transcription factors work in which cellular conditions and what are the actual binding sites which relate to them, the active binding sites, which subsets of genes they work on. And if we can come up with models such as these, then we can, for example, have the promoter region of one unknown gene and then predict what would be its expression profile given its promoter sequence, because we've came up with a model such as this, even if we didn't do the experiment. Or we can understand better the function of this unknown gene based on the model that we've come up with. Okay, so I'll start with a very simple intuitive model of how we come up with models for regulation. So again, the input would be the gene expression. Now, instead of the promoter regions in this model, what we're going to have, so we take the gene expression measurements. This is like, instead of color schemes, we're going to have the actual numbers here. So again, genes are the rows, the experiment are the columns, and instead of colors, we have actual numbers, real value numbers. But instead of the promoters, we assume for now that we actually know the binding sites of the translation factors. So we're going to have another matrix, a matrix which is a discrete numbers matrix. So, for example, if we know that in promoter, the first promoter, we have the binding site of transcription factor number 2, this is going to be like a one entry in this matrix. So we assume we already know the binding sites. And this is the input for our model. So you can think of it that each row here is a gene, and this is like a sample which has both discrete coordinates and real value coordinates. And what we're trying to come up with now is coherent clusters of them. Okay? We're trying to come up with groups which share a common expression profile, but also a common profile in terms of the transcription factor binding sites, which might explain their co-regulation. Okay, so it's like a clustering algorithm, basically. And we're going to use a probabilistic framework to do so. And we're going to use the Bayesian language to do it. So what we have here is the basic model.
I have a question, though.
Yeah.
So in the values in the genomic data section are the identity of the TF?
So I assume I already know the transcription factors. They are known. They're already known, and I know the binding sites. So the entries are like the number of sites that I found for each transcription factor and each gene. Okay, they can be binary, so they can be like, did I find the transcription factor binding site?
But if you have a site, they give you a promoter region, is you slide a window along the
No, what I mean is like I did some pre-processing. I used some algorithm to look for binding sites, okay? And whenever it reported a match, like this transcription factor has a binding site in this specific gene, I would put a 1 in this matrix. That's basically it. Okay, so I know which transcription factors, I assume I know which transcription factors work and which gene. That's the pre-processing stage. It can be binary, it can be discrete values, like the number of sites that you find, 0, 1, 2, 3, up to 4, let's say.
Yeah. The idea is that biologists would look at this and say, oh, yeah, that's solved.
Yeah. We'll see an example in just a second, okay?
So
yeah.
Column, I'm sorry, I still don't get it. What column is associated with a single...
Here, in this matrix?
Transmission factor. That matrix, the genomic data section.
Yeah.
The column there is associated with...
A specific transcription factor, like ABF1.
Okay.
Okay? And all the sites that it found, we found along all the genes in the genome, or the subset of genes that we're looking at. Okay?
So just to keep track, I mean, in your example before, you had kind of interdependence between two different transmission factors, and maybe it's a function of the distance between them or things like that. So you're not modeling anything about
No.
the position of it in this
We'll get into
Okay.
dependencies later. Okay? It's like a first-hand approach. Okay? Okay, so this is how the model looks. We have a Bayesian network here. We have a random variable for each expression in each of the experiments, E1 to En. And we have a random variable for the transcription factor binding sites, R1 to Rk. And what we have here at the top level is like the hidden tag, okay, the model or the cluster of the genes. Remember, we're trying to cluster the groups. This is a hidden variable. We don't know the groups, but we're trying to infer about this hidden variable and trying to find the groups. So what we're trying to learn is both the groupings of the genes and the parameters which govern the dependency. So each model has a characteristic expression profile for each of the experiments and each of the transcription factor binding sites. Okay, so we're trying to find both the parameters for each model and which gene belongs to which module. Okay. Let's look at and of course the attributes here are independent given the module. Okay, so this is more computationally cheap in a sense. Okay, so let's look at an example. Okay, so of course I'm hiding many details because I don't want to get it. I'm trying to get a high level perspective here. There are, we need to use EM for the parameters. We're actually doing structural search because in order for this to work, we need context-specific independence in the nodes. So we need to do structural search, structural EM, weight annealing, and so on and so forth. But I won't get into the details. Let's look at the example of the results. So what we have here is gene expression arrays, which were done in, yeah.
So you're saying you actually have dependency among the...
No, I don't have dependency. I have context-specific independence. It's like saying, okay, let me show the example, and then you'll show what I mean here. So this is yeast stress data. These are experiments done in different conditions of stress for yeast done by Gash and her colleagues. And we clustered the genes. So again, these are the experiments. The tags here are the different conditions, time series in each one of them, different dosage, and so on and so forth. And what you see here, for example, is like cluster 3 that you see it's very high regulated in these specific conditions. And so we come up with these distinctions of these groupings of the genes. You can see that each group has its ups and downs in specific cellular conditions, which makes sense. And this specific group also was related to specific transcription factors like MIG1 and GAL4, which makes sense in these specific conditions. Okay, so what I mean in this context-specific independence is, like if I learn this model, I learn that cluster number 3, module number 3 for that matter, only cares about, it has only a characteristic CPTs, okay, its characteristic parameters only for this subset of these experiments, and for the other ones, like it says, it doesn't care, it has default parameters. Okay, that's basically, that's the basic idea. And if you don't do this, you get too much noise, because if you think of a biological problem here, you have many experiments, okay? But different modules, like different programs of regulatory mechanisms, okay? they don't work in all the conditions. They work at specific conditions. So if you're not going to do context-specific independency, you're not going to get very far.
The idea is there's an implicit second parent of everything. There's an implicit second root node.
No. It's actually, what you do here, what we used here is default tables. So you either have your specific, for a module, you either have specific parameters for this experiment, or you have the default parameters which are shared by all the modules who don't care about this experiment. That's the kind of representation that we used here. Okay?
That also how you invest the problem balancing different types of data, because you're putting all the data in the same sort of reference frame cluster that you have a few regulatory motifs and lots of
right.
expressions, and they're going to be completely ignored in this setup.
Right. So one of the ways you can do this is you can like put extra weight on the nodes that come from the transcription factor binding site and then see the effect. But you're totally right about this problem. We had this problem. Like if you had many nodes from the expression levels that
If you assume that the motif, presence of the motif is a variable that is comparing to the network, then you don't have to do that problem. That's the problem. But then you have to do a structure like
yeah, and that's what you do. Yeah, that's what we do. Exactly. We do structural learning here. Okay. Okay. So let's look at that. Let's get in a higher perspective of what we were doing here. We had this model, and if I simplify it just to get the basic concept, it would look like something like this. So we have a hidden, white means hidden, okay, in our color scheme, module tag, and then given the module, it defines the expression profiles and the regulation profiles, in a sense, which is also hidden. Now, as you can see here, I've changed the color scheme because our model assumed that we knew the regulatory elements, but actually we don't. So this is really white, okay? And there is a good question, which we will come back to later. How does we relate really the sequence into this kind of modeling, so to infer about these regulation events which we don't observe, okay? But the important thing is that, and it relates to some of the questions that were given here, that we weren't getting, so, okay, so whatever you do, you need somehow to get the binding sites of these regulation elements in order to find out whether they affect the gene, yes or no. So you have to come up with these binding sites, and you have we need some way to relate the sequence into the binding sites motifs that you find, so you can infer about the regulation events. And the fact is, we weren't getting so good results as we wanted to, because the identification of binding sites weren't really good. We were using commonly used algorithms at that period, which were not giving us good results. So the first task that we had to handle in order to improve the model is come up with a different or improved way to come up with the binding sites from the sequence. Okay, so the input for this, if we zoom on this specific task, would be we have some prior belief on the common regulation of the subset of the genes, and we have either their, we get their promoter regions, and we try to come up with a binding site model that using it, we would be able to predict new binding sites in other, let's say, test data, either unknown binding sites. Okay, so what are the commonly used binding sites model that we were using at the time? They can be like simple words, okay, over the ACGT alphabet of the DNA with some wildcards maybe, or maybe the IOPAC alphabet, which is like subsets of these 4 letters. Or another representation that was commonly used, still commonly used, is what we call the PSSM, position-specific score matrix. What you see here is the sequence logo of this PSSM. That's the graphical representation of it. And you can think of it in terms of probabilistic interpretation as a matrix where you have an independent distribution over A, C, G, and T for each of the positions. So again, so if I see A, G, T, G, and so on and so forth, I get the score for A at the first position, G, and so on. And then I compute the score. And if it's high enough, I would say, OK, this is a binding site or not. And we will get back to the threshold of deciding this is a binding site or not. We will get on later on in my talk. This is an interesting issue. Mm-hmm. No, that's like basically use here. PSSM is like the scores after you you do the log ratio test using some Markov order 0 of the background, and you get the log score for each of the positions, and you sum them up. Okay? No. Okay, usually. Okay. And the way this was usually done, finding these binding sites motif, like this PSSM, was to look at a subset of genes which you suspected to co-regulate, and then trying to find out like these PSSMs which are common to all these promoters. These are like the blue dots that you find which are common to all these genes in your subset. So it was a commonly used approach at the time, still is. And the problem with this approach is that this might look like a good explanation for co-regulation, but if you would have looked at all the other genes in the genome, like say you have 100 genes which you suspect to be co-regulated, and you look at all the 6,000 genes that you have in the yeast promoters, and their promoters, you would see actually, you'd look at all of them, you see you can find this motif all over the place. So maybe this is common to your subset of genes, but it is common all over the place, so it's not really a good candidate to explain co-regulation, okay, for this unique subset of genes. And what we suggested to do is use a discriminative solution, a discriminative approach in which you try to find motifs which are both common but also unique to the group that you have at hand, which would be something like the red balls that you see here. Maybe it's a little less common, but it's much more unique to this group. And then if you have this motif, you can assess its statistical significance using, for example, the hypergeometric p-value, which asks what's the probability of getting that many red balls in my group if I randomly selected it from all this pool of all these genes with this number of red balls in it. That's basically it. If I was?
If you assume that genes are either random or they have regulatory motifs, then you can assume you can do statistical significance based on just the blue one.
Yeah, of course. That's why the blue ones wouldn't score that high. The problem is that all these approaches usually don't do this kind of search. They don't do this kind of scoring. So they just look for common motifs.
Just based on the length of the motif or how rare it is, you can say whether it's likely or not to have it in the data set. So I suppose that when you're saying these blue ones are appearing all over the place, they are unusual in the sense you can't explain it by random occurrence, but they still appear in all the genes. So what would be the alternative explanation? Why would all genes have the same motif?
Okay. So there are biological reasons for it. There are, like in the promoter regions of yeast, there are low complexity areas called poly-A's, called poly-T's, which are found all over the place. If you would have been, working on motif finding, you would have found them many, many times. And basically, there are. I don't know the exact. I don't think anyone really knows like the exact explanation for finding them, but there are like low complexity regions, very long ones, short ones. And then you would try to find motif, you would find something like, which look like a poly-A, poly-T, which is really nothing.
You think there are some motifs that are useful in a biological way, but you don't care for them for the moment. You care for the ones that are in the gene set and not in the other place. Yeah. I don't know if they are biologically significant, but I know they are there. And they are not good candidates to explain the correlation of this specific subject. Okay, so what we're trying to do here is to extract the relevant bit from the sequences, as we've seen. And like the scheme of what we're trying to do is first we define a search space for these motifs. This can be like the alphabet of A, C, G, T with wildcards in specific length, K of subsequences, or the IOPAC motif I have mentioned. And then, well, wildcard, yeah, a wildcard. Don't care. Okay. And then after you define this search space, you're going to search it either exhaustively or using some efficient way, trying to evaluate all the motifs that you can find in this search space and then score them discriminatively. And the best ones, you're going to evaluate how statistically significant are they and report all the ones that you found really significant or tell that you didn't find anyone. Okay? And let's look at a specific example of how it works. This is the ABF1 transcription factor. We use the targets of this transcription factor according to Li et al. Location analysis, like the experiment that I've talked about at the start.
They have a noisy measurements of whether these transcription factor bind or non-bind to the genes. So we took their measurements, used some thresholds, said, oh, these are the target genes. Let's find if we can find a motif. We came up with this. And this is the actual motif according to TRANSFAC database. This is a database built from known binding sites, which were verified biologically by knockout experiments and so on. So they know exactly the binding sites. They line them together, and they come up with this motif. So even my for-real will say, OK, you got them. OK? So it sounds really great, but it depends on what you're looking for. If I'm trying to say here, OK, I found this is ABF1 motif. This is fine. But let's look at a specific example why I'm not this happy. This is like a specific gene, which Lee et al. Predicted it to be a target. And this is like the area of the binding. And it seems fine, but if you actually look at it, you see that the G here really messed things up because we have an A here. So this motif is like 0, 1. If I don't have an A here, it's nothing. It doesn't match the motif. And you can see that actual binding site model that you come up with from TRANSFAC, you have a low affinity here. So you might tolerate not just A around this position, unlike other positions. Okay? So this is why I wouldn't predict this as a binding site using this coarse kind of modeling, because I don't have any way to describe this low affinities area or the different affinities in different positions, and I can't trade off in a rigorous way sensitivity and specificity when I'm doing the predictions. It's like a 0-1 model. Okay? So this is not very good for genomic-wide scanning, and therefore what I'm going to do is I'm going to do whatever I did before. I'm going to use just as step 1, and then given what I've found, I'm going to do another step, which is refine the motifs, get a better representation of it. Okay? And of course, I want to do it in a probabilistic framework, okay, which I can later relate to regulation and expression. And the way we did it is, again, use a discriminative approach to train such PSSMs. So what we have here, a probabilistic model in which we ask, what's the probability of the sequence being regulated, R being turned on, plus, given the sequence and the model parameters, which is the parameters of the matrix, the PSSM matrix that we've talked about. Okay? And the score that I'm trying to maximize is a likelihood score, which sums up the probability of sum over the probability of regulation over those who are regulated, found to be regulated, and the probability of being regulated, 1 minus the probability of being regulated by the negative group, those who are not supposed to be regulated. Okay, so as you can see here, the score is discriminative because I'm trying to optimize parameters which will increase this term and decrease this term over here. Okay? And the way it actually looks, it looks like logistic regression. I won't go the actual details again, but it looks like a logistic function. We have a threshold value here, and the function here is like, we have the exponent here of the score for each position, at each position in the binding, along the PSSM, and we have some prior. P of I is like the prior of binding and each position along the promoter, which basically means this doesn't decompose nicely, so we're trying to use, we have to use nonlinear optimization to maximize the likelihood here. Okay, so let's get out of the details again, look at the big picture. Okay, what we had here. We had a sequence, okay, and using these formulas that I just described, we have the ability to infer probability of regulation given the sequence and the model parameters, the motif parameters now. So I can infer about the hidden variable about regulation, okay? And of course, I can do it for different regulation events, different transition factors, and use them to try and explain the expression levels that I observed in the experiments that I did, okay? And I'm going to have to use some probabilistic representation of how the expression depends on the model parameters and the regulation event. Now, what we see here actually is a discriminative model which tried to explain the differences in expression given the sequence. So we assume we're given the expression, and the model tries to optimize the probability of the expression given the sequence. Okay, so that's like the first time what we've seen how we relate all three of them together, moving from the sequence up to expression via the regulation events. And then we can try and make if we're at it, we can try and make the model even more complex. We can try and explain the observed expression profile by more attributes. Okay, for example, the experiment type. Like if we did cell cycle phase, experiment type would be the phase in which the cell cycle was in this specific experiment. G1, G2, and so on and so forth. Or we can add another hidden variable. We can try to explain other phenomena in the data that we see in the expression data, which the type doesn't give a good explanation. For example, cell cycle data, which we used here, they had different ways to synchronize the data, and sometimes the cells went out of synchronization. So you might compensate for these phenomena by using this hidden variable over here. How are we doing in terms of time?
Well, we normally finish around 11:30 and then take questions, but we've been doing a lot of questions.
Okay. Okay.
So
Yeah.
The regulation of the hidden variable, the way you describe it, you observe it.
I don't observe it. I infer about it. I use the parameters of the model, the Motif model, to try and infer about what's the probability that this specific sequence is indeed regulated. Okay? And given that it is regulated, I'm going to try to explain, or non-regulated, I'm going to try to explain the observed expression profiles. Okay?
So in absence of EXP, you don't learn anything.
Well, the whole idea is, if you think of the first slide that I've shown, is take what you do observe and try and come up with a model which will explain the observed data in terms of the regulatory mechanisms that are working in the cell. That's basically what we're trying to do here. So we assume we have expression data. That's the whole idea. I'm trying to explain the thousands of measurements that I've seen in a rigorous way, which biologically makes sense. Okay. Okay. So of course, this is going to be duplicated for a single gene. This is going to be duplicated for all the regulator elements that we are looking for, all the experiments that we've done. And the kind of dependency that we're going to see between the expression measurements and the attributes that it is dependent on, we're going to use a presentation which is tree structured. What I'm trying to say is that if I would have done like trying to find out what would be the expression profile for each combination of these variables, which are sometimes hidden, in terms of complexity, this is going to be over-parameterized, the problem, and it doesn't make biological sense. For example, I wouldn't care about regulator number 2 if I'm not in G1 cell cycle phase, because it doesn't work in any other condition. Okay, so learning tree, this is like very similar to decision trees. Okay, learning context-specific representation for this dependency in a tree structure makes biological sense, and it reduces the complexity of our representation. So, for example, what I learned here is I have binary questions here, like is this experiment type G1 phase, and if it is the gene regulated by number 1, regulator number 1, and number 2, and if yes, this is going to be the expected. That's a Gaussian distribution expected.
Yes, it has the first regulatory element in the sequence.
Yeah, the inference.
I thought this is what you were doing before.
Sorry?
I thought this was roughly what you were doing before.
No, what we have here. Well, what we have here is this is hidden. I'm trying to infer about this. Okay, this is not observed.
Whether it's present.
What? Yeah, the assignment to it.
But you have a sequence to know it's there.
No, I don't have given the sequence, I have a probability representation. What's the probability that this is really there? Okay?
So you have probabilistic motifs.
Yeah.
So you are, or you're learning the motifs.
I'm learning the motifs. That's
But I thought
yeah. I'm learning the motifs. That's where.
I thought you first learned them from whether they appear or not.
No, what I'm going to try to do now is I'm going to summarize this. I'm going to learn the whole set. I'm going to learn both the motifs parameters and the structure of the trees and the parameters governing the leaves of the distributions in each.
That analysis you were showing about motifs appearing in the set and not in the big set and so on. You just discard it all, or you
Well, when I start off, I don't know. I need good initialization point, but I don't know.
That's the initialization.
I can use some initialization point, like I can use some educated guess about which genes should have a regulatory element, and then come up with like the basic motif that I've just described, and then plug it in and try to optimize it with the whole set of parameters in this discriminative approach.
So the slide just before this, where you were talking about Lee et al. And so on, that was just for initialization for this.
Yeah, it's like, yeah, in this context, yes. In this context, yes, it's like getting a good starting point. Yeah.
So you're using the known.
But just let me say that you don't have to use like Lee et al. Data. You can use clustering from expression. You can do first do clustering, just based on expression data, and then find motifs, and then plug them together and start refining your model.
What's your sense? Why wouldn't you be able to just do everything with raw data directly?
What do you mean?
Poly A, poly B area.
Yeah, this is like, yeah, you're gonna get a bit stuck at local optimum. That's basically it. You need a good starting point. You can get it from different ways, but you need a good starting point. Yeah. You were saying? Okay. Another data that we had while we were building this model, we introduced it in Recomb 2002. We got hold of really novel data in terms. It was the location data that I was speaking about before. And the way we introduced it into the model, this was like the first paper that held it, made by Itamar Simon, who was then at Young's lab. We used it as location data. That's like the observation that I've talked about, the binding of the transcription factor to the promoter region. But this is noisy measurements. So the way we incorporated it into our model is saying this is observed, that this is like a noisy sensor of the actual regulation events, which we don't observe. Which means that if location data, this is the measurements in location data are like p-values. Okay, they give p-values. So if the p-value is really low, it means there is a good probability that indeed there was regulation. But if the p-value is not so low, you don't get a good score, then it might still be that the regulation event did occur, but you didn't observe it in the data. It's like a noisy sensor. Okay? And then we put it directly as dependent on the hidden regulation events here. That's like the basic model. And again, the technical details which I'm hiding here is we are using probabilistic relational models, PRMs. And so we have to duplicate the objects by gene and experiments. We use shared CPTs for these, and we need to do CSI learning, tree structure, and EM in the model via the regulator variables, so on. But this is like all basic theory, and I'm trying to get a high-level understanding of the problem, and also maybe show you some results so you will be convinced that it actually works. Okay? So we concentrated on the yeast cell cycle data, experiment made by Spellman, and the location data made by Itamar Simon, and yeast promoter regions. Okay, so what we did, one of the tests that we did, I'm not going to show all of them, but this is one of the tests that we did. We hid a fifth of the data. We did cross-validation tests. This is a probabilistic model, so we can measure our ability to predict the observed expression profile over the test data that we've hidden. Okay, so the first thing that we did is like the benchmark is use clustering over both genes and the experiments. So we have the experiment cell cycle phases like the type, and we're using we're clustering genes together, and then using this probabilistic model to predict expression profile. And that's the average log likelihood that we got for held out test data. Then what we did is we added the location data, saying, okay, let's use the data as it is, using a threshold as they use in the paper, saying if it is above the threshold, then I'm going to say this is binding. So location is not a noisy sensor anymore. This is like the actual binding event, the regulation event. We're using the data as is. Okay? And we use this to infer about how the expression profile is gonna look like. And what we actually got is we got worse results. So that's maybe the surprising part. We try to use the data as is, which is proteins binding to DNA, and we get worse results. And the reason is, this is really a noisy sensor. These are noisy measurements. And again, if you don't, so they're using a very strict threshold because they wanted to be sure about their prediction of targets. So if it was below this threshold, it might still have been a regulator regulating some gene, but they wouldn't have observed it. And it would confine our model not to incorporate this data.
This is the threshold optimized? People optimize the threshold?
No, they define the threshold. We use their calls. These are the targets. These are not the targets. Try to use it as is. These are regulated. These are not regulated. Doesn't work. Okay? And then we used location as a noisy sensor of the regulation, but also added the experiment clusters that I've told you about. This is actually white because this is hidden, and we get an improvement in our predictions. And finally, what we did is added the model over the binding sites, over the sequence, and then we get a real increase, a real boost in our performance, which is a very nice result, which basically means that if we add the data, the information that we get of the sequence, we get better prediction performance. So in some senses, we are really coming up with learning to predict the expression profile using the information that we gain from the sequence. Okay? So that's the important aspect. And another important aspect, if you look at this specific work, is like the first time that location data, sequence data, and expression data was integrated into one model together in a unified way, which tried to explain the regulations that we observe in the expression profile. Okay. Of course, as I said, we were also learning the motifs. So these are the kind of the motifs that we were learning for the 9 transcription factors that were measured in the location analysis. These are the ratios of the genes that had this motif found in the group defined by their targets by the location data. This is the expanded group that we defined by the hidden regulation variable that we inferred about, and so on. And you get very good correlation between these motifs and these groups, as you would expect, in a sense. And these are, as I said, the context-specific independence tree structure that we were learning actually enabled us to learn a specific combination of transcription factors in specific cell cycle conditions. These are the kind of combinations that we've learned. Some of them are very well known, like SY6 and SY4, and SY6 and MBP, that's the MBF and SBF complexes, which kick off at G1 cell cycle phase. Some were hypothetical but make biological sense, like NDD1 and MBP here. It's like the kind of combinatorial interactions that we were coming up with. This scheme is like the cell cycle phase, moving from G1 to S, G2, and M. Okay. Again, let's move out of the details. Hopefully I convinced you that we were getting somewhere. And then get a high-level look at the problem we are handling. This is like the basic model that we've talked about here, okay? It's like we have the sequence here explaining the hidden regulation, inferring about the hidden regulation. We have the noisy sensor here and the expression profile that we are trying to explain, okay? And we inferred about it using this model, the probability of regulation, given the sequence and the model parameters. And the model, the binding site model that we were using, is this sequence log of these PSSMs. But what we found out doing this model, that we were still getting many false positives. Too much identification of binding sites, which were not a really good explanation in terms of regulation. And we were trying to think, maybe we can, instead of just looking at the whole picture, regulatory mechanisms. And you can take these kinds of representation, scan the genomic sequence, and come up with many. I wasn't showing you the numbers, but they are really sad numbers. Like you get a very, very high level of false positive.
Not biologically positive.
No. And I would say that using, I'll just show you how we can improve on this, okay? But it's not like the problem is solved with what I'm going to introduce now. I'm getting ahead of myself, okay? If you just look at sequence, you don't incorporate any other data, okay? And you don't take into consideration things that you don't know, like chromatin, 3D structure, and so on and so forth. You're going to get many, many false predictions. Doesn't matter how good your binding site model. If you don't know the exact physics, how it works, the chemical reactions, the 3D structure, and so on, that's what you're going to get. Sometimes you're going to get good results, sometimes bad, but you're still going to have a problem. The question was, can we still do better than this? Okay? Maybe we can use this probabilistic framework to better model this binding sites model, so the whole model that we're trying to build will perform better. Okay? Use the same approach, but zoom in only on the binding sites modeling. Okay? And the specific question we're interested in is this. Let's say this is the promoter. Okay? This is a binding site. Does the fact that I see an A in one position affect the possibility that I'm going to see a C or maybe a T in another position? Okay? Does it has a dependency? Okay? Now, the PSSM that I've just presented, like the commonly used way of modeling binding sites, inherently assumes that the positions are independent. Okay? Now, there are biological reasons to think that sometimes there are dependency. For example, a specific molecule in the transcription factor might interact with two adjacent positions along the DNA, so they are going to be correlated. Okay? But there are two main questions here. One, would you really find dependencies if you would have looked for them? And even if there are dependencies, do we really get an edge in terms of your ability to predict binding sites accurately by modeling them? Maybe this is not a strong effect, so there's not going to be any edge. Okay. So the first thing that we have to come up with to handle this is how to alternatively model binding sites. This is like the sequence logo that we've seen. In terms of Bayesian networks, it would look like this. We have an independent random variable for each of the positions, x1 to x5. This is like a binding site of length 5. And we can suggest other dependency models. For example, we can use a tree structure where we have direct dependencies between the positions. Okay? We can use, so the content of position number 1 is going to affect the content of position number 3 in this model over here. And we can use like global dependencies. This is like a mixture of profiles. You can think of it, this is a hidden variable. So we have subtypes of PSSMs like relating to two different conformations of the transcription factors. So in one conformation, it's going to look for one type of binding site, and in another conformation, it's going to look for a slightly different one. Okay?
So you say P is a hidden variable, but before you told me that x1 to x5 are all hidden variables as well.
No. x1 and x5 are not hidden variables. They are the variables for each of the position of the binding sites. Okay, so you have.
So I guess what I was confusing throughout this whole talk is the relationship between binding site and your RUG variable.
Okay. So the basic idea is that we have a very long promoter. Okay, we don't know exactly where the transcription factor binds to. This was in one of my slides, but maybe I went through this too quickly. What I said in this slide, that we may have some prior belief about where the location of the binding site is, and if we don't, we have a flat prior, like it can bind anywhere. Okay, and the binding site is going to be like k base pairs long. Okay, and we're going to have a random variable for each of the position of the binding site, but it can bind anywhere along the promoters, like a 5-mer, either here, here, or here, you don't know. Okay? And you're trying to come up with, if you have a model for the binding site, it's like defining the probability distribution. If it's just a 5-mer, it's like defining the probability over all possible 5-mers, which is the most probable binding site would be, which is less probable, and so on. Okay? And if you're using a background model, so you would have a background model like the promoter region in general, okay? And then you're going to ask the question, how probable this k-mer that how probable it is that it came from, that's a log ratio test, right? This came here coming from the binding site model or coming from the background model. Okay? That's where I'm aiming here. Okay?
I think strictly speaking, you should have your SC2 variable, sequence variable, as a child node and a parent. You're kind of, your x's are observed.
Yeah.
So you're kind of generating them from a given variable.
I'm kind of generating from...
They're influenced by the given variable, by whether the sequence, you can think about sequence being influenced by whether you have a particular regulator on or not. If you have a regulator on, then you have to have a particular sequence, not quite a particular sequence, but a portion of the sequence should come from a certain probability distribution. You're generating that part of the sequence.
Yeah, I'm going to come up...
X's are kind of from SE2 variable, but that's observed.
Now, what we will come in a few slides will be that what you're talking about is like the model for generating the promoter regions, and you're saying this is like a mixture model, like the observed sequence is either coming from the background or from the binding sites model. But in the binding site model, I might have a hidden variable which is like the subtypes. That's like a mixture model over the binding site model.
You're saying that in your sort of high-level base map, those nodes, when you put in these other something in, the arrows go every which way. So that's probably what's confusing.
Okay, so maybe I didn't explain myself clear enough. Okay, so can we go on? Okay? Okay. And another model what we suggested is like combining these two effects together. We have a hidden variable here, like the subtypes of the mixture, but we have also dependencies like a mixture of trees, basically. Okay? Okay, so these are all very nice hypothetical models, but the question is, do they really do the work? Okay, so the first test would be not look at the promoter regions that you've talked about, where are the binding sites. Let's look at an easier question. I'll give you the binding sites. Okay, this is like aligned samples. Okay, these are very biologically verified sites. This is a very easy questions in terms of machine learning. We have aligned samples from these models. Okay, and I'm going to see if I can get a better job using such models. Okay, so what I have is like aligned binding sites, biologically verified, and I'm going to plug in them into my learning machinery and come up with these different models, and I'm going to see these kind of models really give me an edge. Okay? Okay, so let's look at specific examples first. This is like the profile, the PSSM that was learned for this specific transcription factor using 49 samples, known binding sites. And compare this model to this mixture of PSSMs that we've learned, okay, with this hidden variable node. What you see is the difference here is between here and here. The letters are diminished, and we see here distinctions with two distinct subtypes. This is like 24% of the sites belong to this category, and you see the difference in these positions. This is why it gives us better results in a sense. And if I do, again, test and train over the known binding sites, I get a really improvement, statistically significant improvement in terms of average log likelihood. And this is the tree structure.
Log likelihood is a good measure here. As you're making models more and more complex, you'll have better likelihood.
No, if I'm using more complex models, but I'm testing on test data, hidden out test data, so basically it's going to be fine. Okay, but I think the question is good for another reason, because even if you do, it's still like I'm not going to persuade the biologist that I'm going to tell him, yeah, I'm generalizing much better over known binding sites. He's not going to be convinced to use these models, right? So I'm going to need to come up with different tests. Okay, so okay, that's one example. Another one is the RAP one over here. Again, we see distinctions between the two subtypes and the connections that we learned in the tree structure. Again, improved performance. But you would say, okay, these are very good two examples. What happens if you look at many different transcription factors? So we used like 95 transcription factor data sets which from the TRANSFAC database which have aligned known binding sites. and we compared our ability to generalize better over test data. So that's the improvement in generalization with using dependency models. All the ones that go up means an improvement. In some cases, we had a decrease. And all the blue ones means that the improvement was also found to be statistically significant. Okay? We have a nice way of testing it, but I won't get into this statistical significance. Okay. So data does exhibit dependencies, and I'm not going to get, we don't have much time, so I just want to go back to the high level again. This was like the easier task. We had aligned binding sites. Now the question goes back again, what happens if I have unaligned data? Like long promoters, I don't know the binding site, so I need to learn both the binding sites model, okay, and the location of the binding, both in our algorithm, and come up with these models which will enable me to predict binding sites, okay? And the question, can I do better in this specific task, which is harder, okay? And the test, like the biology test, would be, okay, scan an unknown promoter, okay? Use the log ratio test here to the background, and if this is good enough, you're going to predict this is a binding site. This would be the optimal test for biologists to convince them. We don't have many examples, known ones, like the binding site and all the area which is negatively tested not to be a binding site is a problem. So the solution we came up with is use, again, the location data as a proxy to the truth. We don't have the actual binding sites, but using these scoring, okay, we can say this is regulated, this is not regulated, and compare to the tags that the location data give. Okay, so we do cross-validation. We hide some of the genes, learn the models, and compare the predictions. And if I use different thresholds here over these scores, this is actually, you need a systematic way to do it. And we didn't have any in the literature, so we had to come up with an algorithm to do this, to approximate the p-values of these log scores, which was another paper. Anyway, so if you use different thresholds over the score, you're going to have different trade-offs between sensitivity and specificity. Basically mean you're going to have ROC curves, right? Using the threshold on the scores, you're going to have different sensitivity and specificity, which looks like something like this. This is the profile model that we used for HSF factor binding site, okay? And that's the false positive rate, and that's the sensitivity. This looks fine, but if we compare it, for example, in false positive rate of 1%, these are the other models, which really did much, much better. And again, you would say, okay, this is like the typical best example that you can come up with. How do you do in general? So if I cut it here, this ROC curve, and I compare the performance in one false positive rate, I have a way to estimate when is this point using the scores. And I use it over all location data, like 100 data sets. I come up with this. This is like the delta in sensitivity. This is the delta in specificity when I compare a mixture of trees, models, two PSSMs. So every point over here means we improved both sensitivity and specificity. Of course, to improve just one is not a big thing. I can just move along the ROC curve using different threshold. The real edge is the real benefit is when you push the points further away in both sensitivity and specificity, and that's what we're doing here in most points. Okay, so basically I convinced you this works. Now we can use these fine models and get better predictions about expression levels using the model that we used before, okay? But before we do this, actually, we can test whether this model will give us better correlation to expression without incorporating them in the same model. That's like using the targets that I predict by these binding sites, and let's see how well they correlate to expression profiles when I know the biological meaning of the experiment and I know this transcription factor is supposed to act there. So I expect to see a difference between the targets and the non-targets, okay? So this would be like the histogram of the expression profiles of those who are not regulated, and this would be the histogram of expression profiles of those who are regulated. So there's a big difference here. And there is a question, how do I measure the difference between these two histograms? Because they don't have a parametric form. They are not Gaussians, whatever. So we use Kolmogorov-Smirnov p-value test over the cumulative distribution functions. And these are two examples for the p-values of this test. This is like the correlation to expression that you see when you get a PSSM model for HSF in heat shock, right? HSF means heat shock factor. Okay, and this is heat shock experiments. So you would expect to see a difference, okay? And that's the correlation you see between the targets and the expression. And that's the correlation that you get when you use mixture of trees model, which is much, much better. And that's the correlation when you use the actual predictions by location, which is like an experiment trying to measure this directly, which is the knowledge that we don't have. We use just a binding site model here, okay, to predict targets. That's another example. And getting back to what we did before, I'm going to finish up really soon. I just want to summarize what we've seen here. We had the binding site model, which was the PSSM in this kind of modeling. And what I've basically suggested now is that we can get better results if I replace this kind of modeling with this kind of modeling. That's basically it. Okay? And now, if I want to take a high-level look at all this way of how we model sequence, regulation, and expression together, let's just move location to make the picture simpler. This is the way we model things. And if you go back to the first model that I introduced in the start of my talk, then, okay, I forgot. Okay, there is no module here, right? There is no module. I was talking about modules of regulation, like specific programs. You don't have explicit modules in this model. You don't have them. There is no random variable like module. What you have is like the tree structures that we've learned, okay, like the context-specific defining the expression levels. This is like the modules. They are hidden in this tree structure. They define which genes in which experiments, what's the expression profile, but you don't have in the model. And if you go back to the first model that we started with, this would be like the model. We have a module, and this defines both expression and regulation, which is hidden. Okay, and I asked at the start of my talk, how do I relate sequence to it? Okay, and like the natural answer would be to add a sequence down here, so regulation events define how the sequence is gonna look like. Okay, and all I talked about is like when I improve this kind of modeling by improving the binding sites model, I talked about just the binding sites model. Okay, I said there is background, there is binding sites model. We talked about it. It's also unclear a bit. But actually what I can do is move from binding sites modeling into whole promoter modeling, okay? And try to model the whole sequence, not just like background and motifs, but have a more complex model which can incorporate more biological knowledge and logic. Like, for example, I can put positional bias on the where I expect to find binding sites. I can strand bias, whether it binds to this strand or that strand of the DNA. Structural constraints, like I'm saying, the promoter of a specific module, okay, has these specific transition factors, but you first have to see the first one, and only then that's the right direction in a sense, okay? Things like that. And of course, another important data that you can incorporate into data is phylogenetic data, which kind of can point you to where are the actual binding sites. And there is a very good question, how do you put phylogenetic data, like from closely related species, into this kind of modeling to get better predictions and better modeling? Okay, this is exactly the kind of questions that we're currently working on. Okay. So I'll finish with what I'll start. We had the phenomena that we observed. We try and measure them in different aspects and come up with models that will explain what we observe. Hopefully, I've been able to convince you that we've done quite a way in trying to achieve this. Okay? So did we solve the problem? Okay, if you would have been impatient, you would have said, okay, are we there yet? Did we get there? Did we solve the problem? And of course, being really impatient, you would ask it again and again, like the known figure. And the answer is, of course, not quite, okay? But hopefully, we can enjoy the ride, okay? And that's basically it. Okay. Thank you.
So have any of your predictions been verified?
There were some support to some of the predictions, like the combinatorial interactions that I've talked about. They were supported by other publications as well, but I'm not sure they were supported like in wet biology experiments. Now, the collaborations I was talking to you about were specifically these kinds of verifications. It's like these were publicly available data sets at the time. Location was like very, very new, but we knew the persons involved, so we got the data first. But we didn't have the collaboration that they would go and check it out. Now this is like a different situation.
So I have a comment.
I have a little question. You had a sequence of log likelihood results for your model, and they were using PSSM binding model. So if you were to plug in your Bayesian Net binding model, do you have a log likelihood result you can show for that?
Okay, so I actually, no, I was oversimplifying in a sense, because I'm trying to give a high-level perspective of what I'm doing. I can do this actually, but what I'm trying to do now, instead of plugging, I might do that, but the model that we were using for the PSSM, as I described, it was a really discriminative one. You don't have a background here because this is incorporated into log ratios of each position score. And in the other model, we have like a generative model where we have background and sequence model. So we can still incorporate it in, but I'm not sure this will work that good in this kind of setting. That's why I was showing a different way to relate expression, sequence, is it a discriminative approach? Is it like a generative approach? There are other generative approach used by others, and we are trying to look into them too. I don't have specific results for that. That's the short answer. And what I'm trying to do now is come up with more complex models than what I've shown you here, and use that's exactly what I'm doing now. So I'm using these binding sites model in a more complex model for regulation than what I've shown here. We're trying to figure out which is best. Actually, we're trying to test several ones.
So it seems like you're building more and more complex models. And
what am I?
You're building more and more complex models and taking more and more possible ability into account. And ideally, that's how you want to solve biology. You take all the biological knowledge, and you say you build one model, plug in all the data you have.
Well, you have to be.
But it seems from the description of how you got there, it seems like you don't just run an inference in that model. You do a lot of tricks. So it doesn't seem like one Bayesian really works when you apply. Like the most complex Bayesian doesn't really work on the data. Before that, you do lots of pre-processing, lots of distributed learning. There's lots of stuff that's
Okay, so.
hidden. So I'm just wondering, how much is this really building a Bayesian? How much is it just piecewise statistical analysis?
What do you mean piecewise analysis? I mean.
Well, you have a Bayesian, but then you decide on motifs ahead of time. And then you decide maybe these motifs were not good. So
No, I don't decide. I don't decide on the motifs ahead of time. That's.
Right, you learn them, but you learn them from quite a good initial gap.
No, it depends on the data that I get. One of the benefits that I was showing that I had a pretty good way of coming up with a good starting point, the discriminative function that I'm using. But the starting point may be just take the expression data, cluster it, and then look at the clusters, find out promising like.
I guess my question is, why do you have to do that? Why can't you just from your most complex model just feed in the data and get the result?
Sometimes you can. Sometimes you can. Sometimes, for example, we were using for the binding sites model that I've seen here. Sometimes we use this discriminative seeds approach, which we find promising starting points. But sometimes we were using random restarts and coming up with similar results. But it doesn't work. I mean, it doesn't work always that way. And I don't see it as a drawback. I mean, if you have prior biological knowledge about the problem, why not use it? It's not a bug; it's a feature, in a sense.
Use it, but the question is, can you use it all at once, or do you have to do it piecewise? why can't it be all part of the same model? Why can't, for instance, you could have one model that models all the data that you see, and then, your example, one of your examples was that you're looking for particular motifs that appear in a group of genes but not in another group of genes.
Right.
So you could express that by a model that says that you have a bunch of known motifs that are appear, a bunch of some type of motifs that you're trying to learn, and they appear, some appear in all sequences, and others appear only in sequence sequences. And then you can feed all the data you have and figure this out directly from the model.
Which do you care about?
Right.
Right.
So you could express most of the stuff you talked about. You could have expressed in one big model, but
Well,
you chose and then done inference just once. But you chose not to. So the question is it just historical reason that you're just trying different things out, publishing papers, or is it that actually you can't deal with a problem like this in this way?
Well,
you can't just express all the knowledge that's available and just feed all the data that's available and come up with one.
Well, you can. I mean, there were publications, there were works that they tried to, systematically define all the motifs that they find in the data and then let the model decide, in a sense, which they care about. I don't think the performance was that great. It depends, of course, on the modeling. And if you think of the kind of modeling that we're doing, you're going to have a really big problem in terms of complexity. So you should be doing like an educated guess in how to start your model. And then I didn't get into this, but there is like iterations in which if the motif, that's what the direction you would have liked to go. I mean, if the motif is not good enough, you're going to look for new motifs, come up with new hypotheses about the regulations which occur there, and so on and so forth. So that's basically, that's not like setting them up and then refining them. How many motifs are you looking for? Okay. In the models that I've just described, yes. Yeah. But there are other models, similar models suggested by Aran has worked with me on one of these papers, and in other works that he did, he had like a way in which he decided every once in a while whether he wants to introduce new motifs and so on, which was a good, basically it was like an automation of what we did in this paper, which we did it like in an iteration, but we did it manually in a sense. Okay, so did it in a rigorous way in the algorithm.
How many motifs
Yeah, 10 would be, good. It depends, it also depends on what you're trying to do them. I mean, if you're trying to learn them and all the combinatorial interactions, you're going to have a hard time trying to come up with the models and the parameters for all of them. And one of the ways to handle this, which we are actually doing now, is like you have a relatively large set of motifs, okay? But you constrain the model so that for each module that you have, each regulatory program, you constrain that it's not going to really incorporate more than, let's say, 3 or 4 motifs, which makes biological sense, but it also enables you to reduce the complexity of the model. That's what we are currently doing, and it seems to be giving an edge. Okay, thank you.